Hello and welcome everybody. To those of you that you are in person here at the facilities of Algatec in close to Lisbon, in Portugal, and to all of you that you are connecting online. So today we have the pleasure to invite you to this workshop that is aiming to discuss the best practices on the production and sustainability of microalgae in Europe. My name is Amparo Jimenez Quero. I'm coordinator of one of the projects called co-organizing this workshop, CIRCALGAE project. And together with me, I also have Mariana Doira, which is the coordinator of the MULTI-STR3AM project. And she will explain you a little bit further about the workshop.
Thank you. Thank you, Amparo, and thank you everyone that is here on site, and thank you everyone that is online joining us. So that the next. So we have this joint webinar. We'll have a workshop. We'll have two sessions, two parts with 3 presentations each. And the first part will be from 2:20 now. Starting now until 3:30, and then we will have a short break, and then a second session. This first part will be on talking about the cases of the European project CIRCALGAE and MULTI-STR3AM. We will have 3 presentations. I will ask the 3 presenters to present themselves before starting their talk. We will have a presentation by Luis Costa from A4F, then from Marco Kreis from IGV, and then João Manuel from microalgae in Czech Republic, also in this first part of the workshop. And then in the second part of the workshop, next slide, we will have 3 also presentations, more on microalgae production in Europe. And we will have here then Olivier, Mariana, and Eric to do their presentation, starting on 4 p.m. Lisbon time, 5 p.m. Central European time. So just take this into account. So not taking further, I will pass the floor to Luis Costa, who will do the first presentation of today's workshop.
Thank you, Mariana. Thank you, Mariana, for introducing. As you requested, I will introduce myself briefly. So my name is Luis Costa. I'm one of the executive board members at A4F, Algae for Future. I've been with the company since almost 16 years now. I have a background in biological engineering and in chemical engineering, and I've done mostly technical roles in the company, from engineering activities, large-scale cultivation units, project engineering and implementation, also cultivation activities, biorefinery activities as well, and project management for R&D and for customers. So a little bit of everything, as it is typical in a SME. So this has given me some nice experience in these technological areas, and in the more recent years, I have been able to coordinate different teams inside the company, and now I am in charge of the activities in the technology and in the sustainability areas at A4F. My presentation for today is essentially focusing, of course, on the activities of both the MULTI-STR3AM and the CIRCALGAE projects, and it is focused in the work packages that are devoted to the cultivation of microalgae. So A4F is a project member, a consortium member for both of these projects. In the case of MULTI-STR3AM, we are also the coordinator, but in both of them, we are involved in these work packages for cultivation, besides some other involvements in these projects. So I will both present some insights from autotrophic cultivation, so based on in this case, on sunlight and carbon dioxide, but also on heterotrophic processes, on fermentation processes, because in the multi-stream project, one of the cultivation modes is in fermentation. Next slide, please. Okay, so I will first talk a little bit about A4F, then a little bit about both projects, MULTI-STR3AM and CIRCALGAE, and then I will go into the information that we have been able to retrieve from the phototrophic production of algae, and then also the heterotrophic, and finally some considerations about CAPEX and OPEX reduction results. And the impact of all the technological choices in terms of the sustainability of these processes. Next slide, please. Okay, so first of all, about A4F, for those of you who don't know us, A4F is a group of companies that's devoted to providing technology and know-how in different areas, in microalgae, also in seaweed, in fermentation, and in biorefinery. So these are most of the technological areas that we cover. And we provide services both in developing processes, but also in implementing large-scale facilities in a typical process of TBOT, design, build, operate, and transfer commercial-scale production facilities for algal biomass, both microalgae and seaweeds. So if you want to become an algae producer, you can come to us and we will set up the unit for you and train your team, and you'll be on your way to producing the biomass. And here on the bottom of this slide, you can see a little bit about our track record in terms of microalgae cultivation on the left side of the screen, and on the right side, our shorter macroalgae production track record at pilot and industrial scale. We started out as a company 16 years ago as microalgae devoted company, but seaweed has been in our scope now for about microalgae. So yeah, it's safe now to continue. So the technologies here, we have on the left side two closed production technologies, tubular photobioreactors and flat panel photobioreactors. And on the right side, we have two types of open cultivation systems, cascade raceways and raceways. And for those of you lucky enough to participate in today's CIRCALGAE meeting in the morning, you were able to see some of these technologies in the Algatech Eco Business Park here at this site. Next slide, please. Okay, so about MULTI-STR3AM. MULTI-STR3AM is a project that is devoted to a sustainable multi-strain, multi-method, multi-products microalgae biorefinery, which integrates industrial side streams to create high-value products for food, feed, and fragrance. So you can see the consortium here on the map. There are not so many countries with consortium partners. It's a little bit unusual also for us. So we have 3 Portuguese partners, ourselves at A4F, also IBET and LNEG, which are contributing to the biorefinery processes and also to the sustainability assessment. And then we have Algatech IMIC in the Czech Republic. We'll hear from them later on. And also, finally, in the Netherlands, we have a concentration of the end users for the fractions produced in this project. So we have there International Flavors and Fragrances, IFF, that's taking the fractions to be used in the fragrance industry. Then also Upfield, that is targeting fractions for the food industry, especially in the lipids. And also for farmers, that is taking on several fractions to be applied in feed applications, in animal feed applications. And in the scope of the workshop today, we are essentially focusing on the work package number 2, which is devoted to the cultivation part of the biomass that then goes into the bio refining steps. Next slide, please. Now about the CIRCALGAE project, here this project is devoted to the circular valorization of industrial algae waste streams into high-value products to foster future sustainable blue biorefineries in Europe. So here the scope is a little bit different from multi-stream. Here we talk about already existing biorefinery processes, which are themselves generating some byproducts that can be further valorized into new products. So the consortium is quite bigger and more diverse than the multi-stream consortium. You can see here there is a total of around 20 different partners across several countries in Europe. There is both microalgae and seaweed cultivation going on in this project. However, the focus today is microalgae, because seaweed focus was the first workshop that was going on earlier. So for this project, we have all these industrial waste streams coming from the macroalgae and microalgae refinery processes going, and that we have several project partners that are looking at the characterization of this, looking at how they can be further biorefined and valorized, and also a set of partners formulating these fractions into novel products that are then tested. And of course, all the economic evaluation and market acceptance and sustainability assessment is also going on. So in terms of microalgae biomass production for these two projects, we have, in the case of multi-stream, we have one goal, which is to redesign, install, and commission production units at both A4M and FICOM. So FICOM, I forgot to mention them or to name them. They are a Dutch partner in the multi-stream that is also a producer of the biomass that is used. And FICOM is a producer of microalgae through fermentation processes, so heterotrophic cultivation. And they are producing mainly chlorella strains or chlorella-like strains by fermentation, whereas A4F has the role of implementing processes for the production of nanochloropsis, Tuneliella, and spirulina, essentially in this scope. Then the idea is also to establish optimal biomass production conditions, also optimize the harvesting and the storage of the biomass, and to produce, of course, enough quantities for the remaining activities, the refining activities to go on in these projects. The biochemical composition analysis is always hugely important to determine how these fractions can be formulated into the products. And so here we have, as I mentioned, two partners mainly involved in these activities, A4F and FICOM, and I will present the work of both these partners in my next slides. But in the CIRCALGAE project, the goals here in terms of microalgae cultivation, they are to evaluate the different cultivation parameters to maximize the biomass productivity and also the productivity of the potential microalgae ingredients to be produced. And there are 4 different partners involved in these activities. So ourselves at A4F, but also IGV, also Rocket Klutze, and CEVA in France. So all of these are involved in these processes. Okay, so let's start with phototrophic cultivation. Here you can see some pictures of the cultivation systems used to produce the biomass for in the scope of these projects. So you have here three different strains being considered essentially. So Nanochloropsis, Dunaliella, and Spirulina. You have also three different cultivation technologies being used. In the first picture, we have flat panel photobioreactors, closed reactors, where all of the species, these three species, have been produced. And this is the same also for the other two technologies, the tubular photobioreactors, already at a very large scale. And finally, in the Cascade Raceway, so these are units that are 2,500 square meters each, and that have also been used to produce the three species in the scope of this project. So for all of these cultivations that have been ongoing, there is a monitorization of the growth parameters and of the composition of the biomass that is being produced there. And the focus in this case has been in the most important sources of value coming from this biomass. So, for example, the omega-3 fatty acid EPA, also beta-carotene, carotenoid coming from Dunaliella salina, also phycocyanin, which is a blue protein pigment coming from spirulina, and also soluble protein, just as a bulk extract, also to be targeted. Next slide, please. So here you have these the three different species, but three different species does not mean that there are just three cultivation processes. In fact, two of these species have been produced in two different ways. So the first one, Dunaliella, has been grown under nutrient-depleted conditions and with high radiation, and that's the way to accumulate high amounts of beta-carotene in the cells. So this is targeting the orange biomass that you can see in the picture. But Dunaliella has also been produced without this kind of stress, so with nutrient-replete conditions, and this originates a green biomass, which is richer in protein. So it has different kinds of applications that can be done with the biomass. The same kind of strategies can also be used for Nanochloropsis. And in this case, using nutrient-replete conditions, we have a high accumulation of EPA in the cells and also high biomass productivity and high yields in protein. But if we grow it under nutrient-depleted conditions, in that case, we divert the metabolism, and the accumulation product inside the cells is mainly lipids, neutral lipids, triacylglycerides. So that means a lot of oil will be accumulated in the cells, and that originates a totally different set of products to come along. And finally, for spirulina, this was only grown under nutrient-replete conditions for this purpose, and this means high accumulation of phycocyanin and of protein and soluble protein, which is why this microalgae is best known for. Next slide, please. Okay, so here you have a little bit more detail on the cultivation of Dunaliella in carotenogenic stage. Here you can see the production in a flat panel photobioreactor. And in this graph, here you have the evolution of the biomass dry weight, which is the blue line, and also the carotenoids content as a percentage of salt-free dry weight, and that's the orange line. And then you have also the irradiance, the daily irradiance in the little triangles there. So here you can see throughout, in this case, almost a little bit over 100 cultivation days, how the profile of the culture was evolving, and how the biomass is being accumulated in a semi-continuous way. And then there is harvesting to produce to either whether inoculate another reactor to further scale up or to harvest and collect the biomass. And then in the next cycle, you can see the biomass accumulating again. You can also see that the carotenoid content remains quite high, between 4 and 6% more or less of salt-free dry weight. So it is kept high throughout this process in semi-continuous regime. And on the right-hand side, you have a very simplified diagram for the harvesting and processing here, where the idea is to get the carotenoids extract in oily form, and after that extraction, also the exhaust pellets, which can then be further used for more valorization steps. Well, this is also Dunal yellow, but the color is completely different, of course. This is in nutrient-replete conditions, so you can see the green color of this biomass. And here we have the same type of graph with the evolution of the concentration of the dry weight concentration in the reactor. The highest values achieved are much higher than in carotenogenic phase, because we have to work with high light intensity in the case of the orange cultivation. So here we are allowed, let's say, to keep going for more concentrated cultures. And here you can see also the cycles of harvesting or of inoculation of further scale-up steps along the cultivation dates. Of course, when we characterize the biomass, there are many differences, as I mentioned before, especially in the protein content and the carotenoids content, of course, as well. For the spirulina cultivation, we have here also some of the graphs of the cultivations that were performed in this project. And over on the right-hand side, the main interests here for this production is to obtain a phycocyanin extract, in this case in powder form. So this is a protein extract that's very rich in phycocyanin. And besides that, also the exhausted pellet is obtained, which again can be further valorized. Next slide, please. And for Nanochloropsis, here in nutrient-replete mode, the same activities were going on. Here you can see that the cultivation days, it goes for almost 1 year, and you can see the little crosses that represent the irradiance. You can definitely see the profile of winter and summer there here in Portugal, with the average irradiance per day going in that little sinusoid form. So there you can also see the maximum dry weight achieved in the culture in both times of the year. Of course, we can obtain much more concentrated cultures in the summer period. And here for the biorefinery process, Nanochloropsis is harvested, then the lipid extraction is taking place, and there is again an exhausted pellet that can be further valorized. So one of the most important activities for the phototrophic cultivation, but also for the heterotrophic one, was the integration of side streams in the process. So this took place in several ways. And here you can see here some of the data that was obtained for Dunal yellow cultivation, also for Nanochloropsis. And so typical examples of what has been done by A4F in terms of these integrations are, so in the case of both these species, the integration of a brine industrial effluent, so rich in sodium chloride, as an ingredient to formulate an artificial saltwater medium, instead of using conventional commercial marine salts for this purpose. So to produce the synthetic medium, in this case, it was an effluent stream that was used. And this is, of course, a big advantage not only for the producer of that effluent, but also for the producers of the biomass, which have a reduction in the cost as a result of this. On the other hand, there is also integration of several nitrogen-rich effluent streams into the process, so as a replacement for conventional fertilizers. This has been done with two different streams from two different industries in the vicinity of the algae production facility, and some tests have been ongoing in the formulation, the optimization of the formulation of the media containing these effluent streams instead of using the commercial nitrogen-based fertilizers. Next slide, please. Okay, so in terms of the harvesting done in this in the phototrophic cultivation, so the different technologies have been employed to be able to harvest the different species that have been described. So on the one hand, filtration was used with two different setups. Most of the harvesting activities were performed using a Licoflux system, a large-scale Licoflux system. That's the picture on the right-hand side. This is a cross-flow membrane microfiltration, which is working in an inside-out fashion. So the concentrate is going inside the hollow fibers, and this is a large-scale that exists at the Algatech Activities Park and was used for most of these activities. But there was also another filtration system type, another membrane filtration system type that was employed, and that's on the left-hand side. This is a type of fluidized bed system. It's operating in an outside-in configuration, so it's let's say similar to what a membrane reactor is. So the culture in this case is aspirated into the hollow fibers, let's say, and so this is a different concept that has advantages and drawbacks, and can be useful for certain cases, but for others it's not so advantageous. But this is just the flexibility that we have and that we can test in these cases. Finally, in the middle picture, we have a centrifugation process, which is also is quite different from the filtration one. In this case, it produces a very concentrated wet paste of biomass, which contains almost no moisture on the outside of the cells. So it has the advantage of having less water content for the processes that come afterwards. Okay, so now I switch to the dark side, the heterotrophic production. So these activities have been done by FICOM in the Netherlands in the scope of the multi-stream project. Mainly different species of chlorella and its relatives have been taking place in this project, and this video that FICOM provided us is a good example that one can observe. So this is the inside of a fermentation vessel that they are using, and you can see here the different colors. You can see, of course, the mixing. It's very important. The mixing, the aeration rate, all of these are fundamental parameters to optimize for each process. And here you could see the different colors of what was being cultivated. So these are all chlorella or chlorella related strains. But so you can see the diversity of colors that can be achieved. And here in these pictures in the bottom of the slide, you can also see what the result of those fermentations is, the powders that can be obtained here on the left side. So you see the green one, the yellow one, and the white one. And you also see on the right side the culture stocks that they keep. Also a huge diversity of colors of the the biomass that can be generated. Of course, this is quite interesting potential to be able to fine-tune the composition of pigments and, of course, of other things that go with it for the final biomass. And this enables novel products, but also in many cases, the biorefinery steps that need to be taken can change slightly in order to accommodate the composition of the different biomass types that can be processed. So in FICOM, most of the activities that were done to improve the processes. So here you can see that some modifications, especially in the cultivation parameters, were able to increase by 5-fold the productivity for Chlorella vulgaris and Chlorella sorokiniana in fermentation. So achieving or going up to 2.5 tons dry weight per cubic meter per year productivity, which is quite nice. Here you can also see the capacities for harvesting for these strains that have been achieved at FICOM. So the objective to obtain more than 10 tons per year at FICOM for both of these species in heterotrophic conditions has already been achieved for several months now at their facilities. So at FICOM also some work was done in terms of the harvesting. Here you can see the application of a vibrating membrane filtration system, which brings about some differences from the conventional membrane filtration systems. In fact, the system has been used not only for harvesting of the algae, but it was also used as a means for the preparation of the water and the culture medium to be used. So it can be used to reduce a lot of the disposable equipment that's used for the input to the fermentation. So both air and also water streams. And it is very effective in terms of reducing the contamination of the fermentations. And then in terms of the harvesting, you can see that a very high product concentration was achieved, so 25 to 30% dry weight. So that's quite nice. In terms of the characteristics for the operation, the feedback from the use of this system is that it has very easy maintenance and cleaning and very low overall energy use. So essentially the transmembrane pressure is kept very low because of the vibrating of the system. The fouling of the membranes is very hindered, so it does not happen so much. So it's possible to keep throughout the whole operation very low transmembrane pressure, which means that the energy consumption will also be very low throughout the auto operation. Besides this, this also means that there is no cooling required for the system because of the low pressure that is operated under. And the system is also a sharp cutoff. So it's because there is no fouling formed on the surface of the membranes. There is no dynamic effect over the operation of the actual molecular weight cutoff that occurs. So that means that it's useful in terms of planning the operation because it will have a very sharp cutoff there. And the system comes with the possibility to operate with several types of membranes, both polyether sulfone and PVDF. Those are the possibilities, and you can see it in the picture on the right-hand side that the filtrate, the permeate is very clear. So that's a very good sign. Also enables the possibility to reuse this kind of effluent in further ways. So also the recovery of biomass is very high. This is typical for these kinds of processes. And in terms of the energy costs per kilogram of biomass, investments, etc. So we are talking about similar costs or similar similar effects compared to the conventional membrane system. So in terms of capex and opex reduction, microalgae cultivation and harvesting are still very much associated with high capex and opex, even at industrial scales. Even after cultivation, harvesting and dewatering can account for between 3 to 15% of biomass production costs. So it's still a significant part, but the cultivation part is typically the most demanding one. And depending on the type and location of the unit, the OPEX can represent more than 50% of the biomass production cost. Of course, this is hugely variable because there are no two identical algae production facilities. But of course, to ensure that these products are economically attractive, both A4F and FICOM are introducing design improvements to these two steps to reduce capex and opex. In the case of phototrophic cultivation, OPEX reduction was achieved by employing culture medium recycling, and this decreases the costs with culture medium by 78%. Then also integrate the water management with nearby industries, which is a very good advantage of having industrial synergies in an industrial ecosystem, as is the case here. Also optimization of the growth conditions, because of course every time we optimize the growth conditions, there is a better use of resources. Then also increasing the robustness of the management processes and the data analysis of the results to improve the performance. Installing additional sensors and flow meters to correctly measure all the inputs and be able to then act upon the information that is collected from that. And the final point to introduce side streams that are coming from other industries as feedstocks, so that enables the reduction of some costs. In the case of the brine that I mentioned before, we are talking about a reduction of cost by practically 90% in terms of the water and marine salts associated to the production of the culture medium. Besides the OPEX reduction, also in terms of optimizing the operation conditions, these are still activities that are going on with the continuation of the project. We talk about the optimization of the temperature control of the reactors and also reducing the levels of dissolved oxygen to improve the productivity. In terms of capex, this can be accomplished in terms of the heterotrophic processes. So now talking about the input from FICOM. So scaling, of course, is always a typical way to achieve this reduction of capex. Also, alternative processing strategies. So, for example, using the vibrating membranes is a way to introduce that. Also, FICOM has combined two steps that they had before, drying and milling. They are now combined in a single technology, so that also reduced the capex. And so these are the main areas of focus in this case. For OPEX, the maximization of the efficiency of conversion improved the density of biomass in the reactor. So this improves a lot the efficiency of the consumption of electricity, for example. And they were able to reduce the cycle time between start and harvest because of enhancements in the productivity. They have also been able to automate the processing steps, which also reduced the downtime for those steps. So this is my last slide with just some considerations about the sustainability improvements that derive from the work in this work package 2. So the three main areas here of focus. Number one, the utilization of the side streams. So this is on the one hand decreasing the impact at the origin of these side streams. So it's it's taking care of a problem that is typically a source of lower sustainability in their process. Then, as is the case in the utilization of brine, there is a reduction on the consumption of water and marine salts for the algae cultivation activity, and also in the case of using nitrogen-rich effluent streams, reduction in the consumption of fertilizers, and in the case of FICOM, in terms of using alternative sugar sources or carbon sources for the fermentation processes, also a reduction in the conventional sugar source consumption. So this is the first main topic, the utilization of side streams. Then the second one is to intensify all the processes. So working with higher biomass concentrations in cultivation and downstream. So in FICOM, it was shown a five-fold improvement in this. And so this means that the energy consumption per kilogram of product is reduced dramatically because of that. Also the water consumption, you have the same water used for one fermentation, but five-fold higher biomass there. And then, of course, also the reduction of the infrastructure impact, because the same infrastructure will have 5 times more productivity. Then finally, also the same kind of impacts, but from improvements in the productivity of the system, not just working with higher biomass concentrations, but also having each day the production or the productivity of the biomass being increased is also a way to utilize the resources in a more efficient way. Next slide, please. I think this is my last one, but just to check, it is. So thanks a lot. I'm able to take your questions. I think at the end of the session. Yes. Okay. But thank you for your attention.
Thank you, Luis. I will now invite Marco from IGV for his presentation. Ask you to present yourself, and then I'll ask everyone also to keep the questioning in the chat that we will follow with the Q&A after, and what we're not able to respond now, we will follow with by email with the answers. Okay.
Hello, everybody. My name is Marco Kreische. I cultivate microalgae at a German company, IGV. We're located near the city of Potsdam, near Berlin. And I worked there since 2018, directly after studying biochemistry and molecular biology. So IGV is producing mainly extracts from microalgae, but also plants and herbs for cosmetic applications. More than 90% of our extracts are for cosmetic applications. We have a known production of microalgae biomass. Usually we produce only those biomasses that are easy, not so easy to buy on the market. We also use biomass for partners. Or that is too expensive, we cultivate ourselves. We don't have these huge capacities like they are here in Aldi Future, but we cultivate ourselves. Beside this, we have also known cosmetic manufacturing for a wide range of cosmetic products, and we sell them online mainly. And we do research projects to learn more about other algae applications beside cosmetics, and also to develop new extracts for our cosmetic customers. So we also have 2 other departments that are not dealing with algae in IGV. So one about analytics of feed and food products, and one about food technology and bakery technology, which are beside the algae department. Next slide, please. So I just want to mention that when I speak about microalgae here, I include cyanobacteria. I know some people see it a bit different. Just to make it simple, I speak about microalgae, including cyanobacteria. So also when I speak about what is special in IGV, what many other microalgae companies don't do so much, is that we are kind of specialized in growing non-food microalgae, so not the classical ones that are in use for food applications and that are approved by food regulations, but novel, also cyanobacteria like Mastocalus and others that we cultivate. We cultivate diatoms like Pheodactylum that is in the CIRCALGAE project, but also freshwater diatoms. We cultivate red algae like Cyonidium, Galderia, and Porphyridium. Also everything for cosmetic applications, and also many green algae, Tetradesmus, Synodesmus, Anucleopsis, these things we do also mainly for cosmetic applications. What is also special in IGV is that we not only have tubular photobioreactors that we mainly use for cultivating these different algae, and that we produced till the year of 2013 in the company, then stopped it. We have also an experimental type of photobioreactor where the algae are dispersed in smaller droplets, like you can see in these photos, and they grow with a high surface for gas exchange and light. But this is an experimental system. Most of the biomass that we use comes from tubular photobioreactors. And since we constructed till 2013 these photobioreactors, we have prototypes from that time that are still running, and they have a lot of different tube diameters, pumps, different characteristics that we can play with different algae, what is working, what is not working. So this is a big advantage that we have different systems to try for every microalgae. And another thing is that since we don't cover all of our capacity by our own production from biomass, we have to buy biomass from partners, and we get in touch with the real prices of microalgae that are on the market in our range of about hundreds of kilograms dry weight usually take. So you can see in the next slide on the vertical axis, that's the price per kilogram. And in the diagram are all the most discussed microalgae applications today. And what we see is today we see microalgae already in cosmetics, in food supplements, and food additives, beverages, things like this. And the cheapest microalgae on the market, they already scratch a little bit on these pet food and food applications, but there are many much more beautiful applications below this red line at the diagram that we would like to see on the market, not only see them feasible in projects. And what we have to do to see them is to lower the price of microalgae to actually see them on the market in our hands of the customers. And this is a real big thing in microalgae industry to lower the microalgae prices. For sure, there are some regions of price where there's a long way to go, and also it depends on alternative technologies, how they develop. We saw, for example, that solar panels and also electric cars, they developed much faster than the microalgae price was lowering. So for this biofuel and carbon capture segments, we are still quite far and went even far from being in a commercial situation. But all these other applications in the middle of this diagram, they should be our focus for the next decades in our lifetime, to see them, to have them in hands. Yes. This is again the red line, just to remember that this is not what we, IGV as a company, is reaching these prices in our scale, since we produce hundreds of kilograms. And this red line, what really brings us to the big commercial applications, it's what's reached in hundreds of tons scale dry weight in big plants. Like there is one example on the right side, which is producing 700 tons per year. This is what are the cheap microalgae that I'm speaking about, that we can use in future for many other applications besides cosmetics. Yes. And as we see this red curve, if we have production below 1 ton per year, usually we see this personal cost fraction in the cost structure is making the cost exploding. But we see the higher we go in the production tons per year, the less important personal costs are becoming, and we have more of these construction equipment questions and also energy costs that are there are the reasons for the curve not to flatten as much as we would like to at the moment. So we need to go very much upscale in future. This is one of the biggest cost factors that has to be overcome. And for the next classical ones, I go to the next slide, which is like just an easy model of the input and output of microalgae. So in this model, the output, like tons of dry weight per area per year in the photobioreactor, is represented by the volume in this barrel made of staves of different cultivation factors. So every stave is representing a cultivation factor in this case. And there's always one limiting. And in this example, for example, the light is the limiting factor. And we see on the right upper diagram, we tried to overcome this during the winter in Germany. We tried to keep a PBR running also in winter when the light was completely too low to grow at a certain growth rate. And yes, it's possible that we could use the PBR all the winter. We did not have extra cost for PBR itself since it's still there. So this is one way to increase the output, but for sure it comes with at a certain price, in this case with a high energy demand, which is one thing to solve this in future by alternative energy sources like photovoltaics or also other wind, solar energy, geothermal energy, which we will also later on here, which is possible to increase the output side. The other thing in this diagram is that to reduce the input side, we could cut all these staves to a size that is not necessary. Like these non-limiting staves can be reduced. For example, this mixing, which is also requiring energy, in this example could be adjusted to the light intensity. Also, the salt content in this case is not really necessary for the algae to have this 30 gram per liter. It could be also 10 gram per liter. And this is exactly what we did, for example, in CIRCALGAE with our Phaeodactylum. We reduced the salt content of the medium and also tried to do medium recycling. At the lower diagram on the right side, we can see just an example of a saltwater algae that is how much input from material side is needed for 1 kilogram of algae to produce. So if we do not recycle salt water with 30 gram per liter, and we grow our algae to a maximum of 87 gram per liter, we will use in the end like something like 5 kilogram of salt for producing 1 kilogram of algae. So we need these technologies to recycle the medium, to also lower the salt content. This is also helping, but not only the salt, but also other medium components, the water use, the CO2, for sure we need, and not everything is ending up in the algae. And for cleaning of the PBRs, these are typical material inputs, just as an example for not optimized process. Because for IGV, since we cultivate a lot of microalgae that are quite new, we rarely have a very optimized process. So we just to make basic changes in the beginning, usually to keep them growing and cultivation running. So then we can go to the next slide. Like this is an example of one of our PBRs running with Phaeodactylum tricholutum, which we produced for the CIRCALGAE project. So we applied to reduce the salt content in the medium. We saw that we had some contamination problems with this algae, and we tried to overcome it with using the algae's ability to grow at high pH, like it is also done for spirulina, for example. So we managed this algae to cultivate at pH 10 It's not a problem for the algae. However, it brings some other smaller problems with it, like carbonate, precipitation things. But in principle, it's possible and we can do it. And also we saw that the chlorophyll content of the biomass was influenced by the light per biomass. So we were able to optimize this a little bit by shading the days before harvesting. And in the diagram, you can see also the fractionation of the biomass that is usually done by CO2 extraction. So we have this lipid extract, which is the main product on the one side. And as a side stream, we have the dry, defatted residue after CO2 extraction. And this is, in our case, used for the CIRCALGAE project to develop new products, to have a whole use of the biomass, which is, this biomass is very valuable and we can't afford to throw out of it. So we have to have a complete use in the future to be cost efficient in this case. So, and on the next slide, just to mention why we improved this pheolactum cultivation. So on the one side, we have this wonderful fucoxanthin pigment, which is in pheolactum, which is promising for food applications. We also have icosapentaenoic acid, which is a fatty acid. And also pheolactum is a promising model diatom for future production of other things via metabolic engineering, for example, antibodies and more. And the current price is about 150 euro, which is preventing some applications also as a food supplement. And there is also a history of using pheolactum in food and other applications. So about year 2018, must have been, two companies made novel food applications in Europe for pheolactum, for not the whole biomass, but for extracts from it. And just last year, the EFSA made a decision that these extracts are not allowed for novel food use in Europe, because the cultivation needs to be optimized. That was one of the reasons. So it's quite an actual thing to keep a consistent and safe production process for pheolactum, which is needed to bring it on the market later. So, however, also the CIRCALGAE consortium did a great job and published a deliverable, which is public accessible, where a lot of detailed information about this, also about the whole industry, not only pheolactum, also about other microalgae. And it's also linked on the CIRCALGAE website. So we can go to the next slide. And this is about what I think will be the future of microalgae production, not only in Europe in general. So if we have a look to what was done in the past of other crops, not microalgae, but classical crops, we can also see what happened in the last thousands of years. And if we see it in this table, for example, 3,500 years ago, people cultivated the first time tomatoes, and they were so small, it's not worth to grow them. But they improved these strains, and the yield became higher. And today, you see them everywhere, and everybody's using them, and they are produced in billions of tons in Europe. And this is what I think is also the perspective for microalgae. Even if we are not at the moment at the yield of a tomato greenhouse, would have been great for the people that 3,500 years ago tried to grow tomatoes. They could have imagined what beautiful greenhouse we have now without soil and without anything. So we can't imagine very much what is in some years with microalgae cultivation. But for sure, strain improvement will be the next big thing on the long run, which we will hear also in the next presentation then. So we will have, we already have not green chlorella like these golden chlorella, these white, yellow colors. So we already improve strains, not only by cultivation conditions, but also by changing the strains. but there will be other tools in future also, including metabolic engineering of new pathways. For example, there could be in some decades pheolactodum, which is temperature resistant, which has like corn today, some resistance genes that is protecting them. We can have not only 1% glucanthine inside, maybe we can increase this to 10%. So just as an example, this will be the next big thing that I think we have with microalgae, like with all the other crops too. And the second thing that we also can learn from history is that all these cultivation systems and the methods for growing and harvesting and processing, they will become very species specific and product specific. So at the moment, we cultivate more or less many microalgae with tubular photobioreactors. We already see some specialized systems used for this linear cultivation, only ponds, because it's the cheapest. Like there is already diversifying, but we will see in the end there will be a production system for every product, for every species. Like we have now, even if, for example, tomatoes and potatoes are closely related, we have completely different cultivation systems. This we will have also for algae. So this is what I expect then. And we will for sure use also massive energy in cultivation, but from renewable sources like geothermal and solar energy. We will keep coupling the production with sources of pure CO2. We will not do this carbon capture storage things, but for sure we will try to use CO2 where we have it anyway. And also heterotrophic production will become big, since maybe we can introduce also heterotrophic pathways in algae that are today only phototrophic in some years. So it's a far way to go, but these are all objectives to think about in future. And since the areas are limited for cultivation of other crops, we also should have in mind that we produce the microalgae on areas that are suitable, like on sea surfaces that are mainly unused at the moment, not only on barren land, but also on facades of buildings and other places, maybe on Mars, we will see. So we will find a place for the algae, and there's a lot of ocean area left. I think this will be also a big change in some years for this. So I finished. This is my last slide. And I give the word to the next speaker.
Well, I'll now invite João Manuel, that is joining us online. If you could share your screen and present yourself, please.
Hello, everyone. Hello, everyone. My name is João Manuel. I'm a PhD student here in Algatech from the Microbiology Institute in the Czech Republic. I've been conducting here my PhD work. I'm enrolled in the University of South Bohemian in the Czech Republic. And I'm going to talk to you about the improvement of microalgae strains and how does it work. So at first, why microalgae? We all know they can provide high yield. They have a lot of interesting products that cannot be obtained in other systems, or even they can be obtained in other systems, but with microalgae it can prove to be more productive. It's sustainable. Especially with autotrophic algae, they can sequester CO2, and a few years ago there was also some hype with biofuels. So, with what we want with the strain improvement, we want higher growth rates and biomass yields to to enhance specific productivity is, of course, from lipids to proteins to pigments, increasing also tolerance to stress conditions. Which steps do we have to take into account? First, we do a strain selection, and also we have two main steps: strain selection and then the improvement strategies. In terms of strain selections, of course, we can take into account lipid, for instance, the lipid profile, pigment content, and growth conditions. Is it a species that grows well in the Portuguese climate, or is it a species that grows well in the Swedish climate, for instance? In terms of improvement strategies, we could distinguish them between abiotic and biotic. Abiotic, we have temperatures, light conditions or photoperiod, light more intense, light less intense, pH. Yes?
Can you hear me just a moment, because you are in full screen, but the screen that we are looking at is your support.
Okay.
Share the screen or share the.
Yeah. So let me see again.
If, sometimes it happens.
This one. So now it's okay?
Yes, now the full screen. If yeah, but it's I think you need to
First full screen and then share.
Yes, first put in full screen mode and then share.
Okay, wait. No, stop.
Stop. Put in full screen mode and then go to the Teams, maybe.
Okay. Go here. No, I'm having troubles with it.
Yeah, but
Display settings. No, maybe. So, no. How is it now? No.
No. I don't say anything. It's because maybe you have to share. Said the window, but the screen, not the window, or vice versa. Now you go to. Yes, fantastic.
Now it's okay. It's working.
Yes, perfect.
Perfect. Okay. So.
Yes.
Okay. Yeah. Okay. So continuing. In the biotic parts, we could have the biotic genetic approaches. Straightforward genetic modifications, for instance, and also co-cultivation approaches has been gathering more attention in these recent years. In genetic approaches, we have could have random mutagenesis, and for instance, CRISPR-Cas. And overexpression, heterologous expression. In co-cultivation approaches, we could have algae with algae or algae with bacteria. Also possible with algae fungi. So if in terms of practical examples, for what we have been working here as part of the multi-stream project, first, I'm going to talk about the microalgae, as an alternative to palm oil. Palm oil, it's mostly comes from the palm tree, cultivated in South Asia, and sustainable so that it does not lead to deforestation for the cultivation of palm tree. So we have to find the strains that can provide that can have a lipid profile, a fatty acid profile close to the palm oil. We did the screening, and we selected these 3 strains, for they were the ones that were showing the closest profile, it amounts specifically palmatic acid as compared to the palm oil. These were the three strains, Desmodesmus and two Chlamydomonas. Two Desmodesmus strains and one Chlamydomonas. They're normally cultivated with BG11, and we did the optimization first in small scale, and then after that we finally cultivated them in our 30-liter photobioreactor, which we designed here in Algatech. And we applied the stress condition, in this case, salt stress. These values here I didn't put here, but they are what was already available in the literature. I can say that we applied, in our case, 16 grams per liter of salt stress. And then these were the results. With this strain, so here we have the BG11, which is the normal media that we use, and then here the salt part is what it refers to when we applied the salt. The growth was, so the salt was applied from the very beginning of inoculation of the photobioreactor. This first strain, the culture crashed after 3 days, so it could not deal with the stress of the salt. And the other two presented good growth, being this strain 463 presenting a good final dry weight, and also in terms of total fatty acids, also a good amount. They will have to be, the idea is to continue carrying these experiments to further fine-tune this optimization. Then, continuing on the abiotic factors, we're going to talk about microalgae as a source for lutein. Lutein is it's mostly sourced from marigold plants. It gives 2 cultivars per 2 cultivars. It's not cultivars, I'm missing the term, per year. I worked with these algae. It's a green algae, Monoraphidium. It was collected in the Antarctica by a team from the Botanic Institute, also from the Czech Academy of Sciences here in Chamonix. It's capable of accumulating high amounts of lutein, about 2.6% of its biomass. This was in a raceway pond back in 2020 that we were able to obtain. Usually in the laboratory, I've been in terms of control, it has been around 13-14%, but we managed to obtain in a raceway pond about 100 liters, if I'm not mistaken, 110 liters. We were able to obtain these concentrations, and so it comes from the Antarctica. It's not the species a sacrophilic algae, which means that it's only adapted to cold temperatures, but it's actually cold tolerant. We found out that it's cold tolerant. It grows well between temperatures between 10 and 20 degrees. Above that, 25 degrees, it's already suffering a lot. These were the conditions tested. So the control at 20 degrees with CO2 supplementation and continuous light. And so far the conditions tested just air, so no CO2 supplementation, with a light dark cycle, and then even lower temperatures. And these were the results. We can see that with no CO2 supplementation and with a light dark cycle, the growth in terms of biomass of dry weight was lower than the control. but here with the 10 to 15 degrees, it was possible to obtain higher growth. In terms of the lutein cellular content, the air production, so the no CO2 supplementation was the lowest one, and it was with a light providing a light dark conditions, it was the highest. It was comparable then 10 degrees and 15 degrees to control. But when we take into account the dry biomass that was able to be obtained, we could achieve higher productivities of about 29% at 10 degrees and then 21% at 15 degrees. This again part of the part of the multi-stream project. And then the biotic factors. Here I've been working with this algae before for my PhD, and the idea here is to type into the microalgae's phycosphere to see how can that improve our productions. So I first grew a small sample of the algae culture in LB media. It's a rich media for specifically aimed for bacteria. Through numerous pests, I inoculated plates, I isolated, and then analyzed the supernatant of the bacteria through MS analysis. And then I was able to construct, with the help of Kumar Saurev, we constructed these network maps where we were able to detect specific molecules inside in the bacteria isolates, and also the algal supernatant, that molecules that are related to quarant sensing. And what is quarant sensing? It's a well-known mechanism of communication between bacteria, where they keep releasing into their surrounding media these signaling molecules, and when they reach a certain quorum, so high concentration in the media, these molecules that keep being excreted and also absorbed by the bacteria, they elicit a response. That response can be from movement to a different area, to releasing exopolysaccharides, to releasing algicides, and much other molecules. So then I selected the bacteria. I did some preliminary studies. The bacteria, isolate was then selected, which was then cultivated at higher volumes. That same supernatant, again, this is the supernatant without the biomass of the bacteria, was extracted and concentrated and then applied to the different experiments performed. So then first we have a concentration optimization. These amounts refer to the weight of the supernatant, the concentrated extracted supernatant. And here we can see that 40, in this case, the highest concentration of the extracted supernatant, sorry, the 20 micrograms per milliliter of this supernatant was the one showing the most comparable biomass growth to the control. And then here we can see that it was also showing the highest lutein amount in that culture. We then continue to a higher volume, and here again reaching. It showed in the beginning, in the middle of the experiment, a slower growth, but eventually showing comparable growth to the control. And then in the end, in the last day. This treated culture with the bacterial supernatant showed a 41% improvement in lutein content, in the cellular lutein content. This is a different approach of growing, taking into account microalgae and bacteria. Normally, or most works are focused on getting microalgae A together with bacteria B. In this case, the idea was to work with the relations already pre-established in the phycosphere of this algae, and we showed that it can be used for biomass improvements. Then, continuing in the biotic factors, we have UV mutagenesis as an example. Here, we're also talking about the work being done in this project, multi-stream, where also for lutein production, where we have the chlorella growing heterotrophically, and then through that it has chlorophyll, obviously, chlorophyll and carotenoids. And after applying the UV treatment, a number of mutants were able to be obtained with all with different characteristics. But the most interesting ones are here, where they show no chlorophyll, and it's mostly carotenoids that they present. And then we have here an example already with this strain. This is an old strain that it was obtained here all through this type of mutagenesis, that it can produce a high amount of lutein and without chlorophyll, which makes it easy to extract the lutein, not to extract the lutein per se, but it's easier because then you have the lutein, just the lutein, and not the chlorophylls. These other mutants, of course, will be, specifically this one, will be continued to better optimize these productions. This this one regarding also the yellow, that other this mutant over here. This would then we have again to apply the abiotic factors to further optimize its production. We can use this 4-factorial experiment using several factors at the same time. In this case, glucose content, nitrogen content, phosphorus content, and also temperature. And then through this, we can easily get to the optimal conditions to growth without specifically testing necessarily. Imagine you have glucose content, instead of testing 0, 5 grams, 10 grams, so on, you can, with this in this way, test only a few factors, in this case regarding glucose, and then through a mathematical modeling, you can reach to the optimal amount of glucose. Then in the biotic, continuing, we have targeted genetic modifications. Here I'm going to talk about another project that it's now part, the Algatech is now part of OPIAC. And the idea here is to produce therapeutical peptides in cyanobacteria through phycobilisomes. And the idea is with applying metabolic engineering, these will produce amino acids that can then be used for these therapeutical one of the parts of this project. So this is one part of this great project. With it will be focused on Synecocystis, where we have a multiple array of genetic tools available in Algatech, specifically CRISPR-Cas using plasmids and homolog recombinations. And also this algae is also capable of growing heterotrophically, then able to yield higher productivities. The idea here is that to have then this organism, this modified organism that can give us these new therapies. And then how to get the question, going back to the multi-stream project, how to get these metabolites. And then, so the Jose Gil, which is part of our group, he works with counter current chromatography. Here, these are the principles of this technique. I won't go much further into this, into the principles of how it works, but basically it is a helical column rotation that generates a pumping force through the Archimedes pumping action. With this method, it is possible to automatically, in this case lutein, to automatically extract a pure form of lutein, and all the solvents used can be recyclable and used and reused. This is another example with for the extraction of EPA, and where he and Daniela Barcenas were able to reach 99% of purity. So the take-home messages: the optimization of these strains, it's important so that we can reach better productions, and also then for that allow for the expression of these valuable metabolites. There is out there high diversity of algae species that can be used, and that it's possible to use to better maximize this productivity. And the combination of these biotic and abiotic techniques has to be considered for these enhancements, and also very important to consider what is the end goal of this optimization. If we want, for instance, if we want to use this microalgae for food applications, we cannot use target mutagenesis, the so-called GMOs. In this case, we have to consider UV mutagenesis. It's random. This is okay, but if we go specifically there to change the genome of the algae, that cannot be used. Then it cannot be used for human food. Thank you very much. These are my colleagues that worked on these projects, and thank you very much.
Well, thank you, João Manuel, also for your presentation. And we will make a short break, half an hour break, and we will be back to the second session. We have some questionings from the chat. I also asked who is doing the questions to send them their emails as well. We will continue after with the second session, and if we have time, we will address these questions now. Otherwise, after the second session, otherwise we will send them the answers by email too. Okay, to not move further, delay further. Okay. So in half an hour, we will be back. Thank you. Okay, so let's start the second part of this webinar workshop. We are going to start the second part dedicated to microalgae production in Europe with 3 speakers, 15-minute presentation. Please be on time on the presentation to leave time for question session. Olivier, Mariana, and Jean-Eric, if you agree, we can pass your slide to avoid to share your screen. But if you have preferences, we can also let you share your screen. So let us. But all the presentation have been compiled, so as you want.
So I have some animation, so maybe I will try to share my slides. I mean, if it's not working, whatever. But
okay. So Olivier is going to start. And Olivier is from Fermentalgue in France. He's going to talk about phycocyanin production by Galderia sulfuraria and side stream valorization. Let's start, Olivier.
Okay, thanks. First, I would like to thank the organizer for giving me the opportunity to do this presentation and what we are doing also at Fermentalgue. As you said, I'm going to talk about phycocyanin production and with the microalgae Galderia sulfuraria.
Excuse me, you are presentator mode, and so we have your slides in, the first one and the second one, the sharing screen.
It's
Okay.
close the presentation, then stop the sharing, then on your computer, put in the presentation mode, and then share the screen.
So you.
Now in your stop the yeah, you already stopped it. Now in your computer, put in the PowerPoint in the presentation mode.
No, but share it for me. It's going to be easier. So anyway, so
Share the window.
No.
No?
Por aquí.
See.
take over and share the screen.
Yes, it's.
I will.
No problem.
So what?
Ça parle pas. Donc, il est en train
Share. And then take. Okay, you can start.
Okay. Yeah, I don't see the slide.
It's being shared in the Teams.
Yeah, perfect. So you can go to the next one, please. So that's the main project that we have at Fermentalgue now running. The first one is on DHA. DHA oil from omega-3 from extracted from Schizochytrium Trostochitris strain. Sorry, that's actually under commercialization product, and we are producing that product in Europe. The second project is the Blue project, which is in pre-industrial phase. Objective is to produce the phycocyanin as a natural blue colorant and also for nutraceuticals and antioxidants. And this project is a joint development agreement with the Givaudan company. And third is the spin-off of Fermentalgue that has been done with the Suez Group. That aims to develop equipments for the culture of microalgae and the carbon capture on autotrophic conditions. So it's actually running, and yeah, they're trying to develop this photo bioreactor systems. Next slide, please. So at Fermentalgue, the main technology we're using to grow microalgae is the bioreactors, so fermentation. we decided to select a strain that was able to produce phycocyanin and able to be fermented. So unfortunately, spirulina cannot be used for that kind of cultivation tool. And what we did is that we looked in the literature, what organisms has the capacity to produce phycocyanin, and also being able to be produced in fermenter. So after a quick review, we found out that the specific strain Galdiaria sulfuraria that belongs to the green algae has this capacity, and other the Galdiaria sulfuraria is an extremophilic strain that grows at acidic pH and high temperature that are found in hot springs and also volcanic area. Galdiaria also have the advantage to be grown in mixotrophic and heterotrophic conditions. And it also has the advantage to consume a lot of different carbon sources to enable its growth. From what we had in the literature and what we experimented also at Fermental is that the other advantage is the productivity in terms of biomass, that 50 to 100 times more than the spirulina, and also in terms of phycocyanin. So 20 to 30 more phycocyanin productivity compared to spirulina. So in the picture that you have you can see the strain itself on the left with the chloroplasts, and the green-blue color, which is a mix of chlorophyll color and phycocyanin. Phycocyanin being a photopigment like the chlorophyll. When you grow Galdiaria in mixotrophic conditions, of course, you have the induction of the genes and the production of chlorophyll and phycocyanin. However, when you grow it in the dark, you can sustain the growth with the carbon, but as there's no light, they cannot produce phycocyanin. So, which was for us an inconvenience to produce phycocyanin. So, next slide, please. What we did is, okay, that's not the same. So, what we did is we did some strain selection at first. And we succeeded to select a strain that was able to produce phycocyanin in the dark. And we did like the regular breeding for plants. We did selection and again over the same strain and over. And we've been able to achieve a good yield of phycocyanin in the strain, equivalent to the one of spirulina with lights. So from that, we develop a fermentation process based on glucose in sterile conditions, without light, of course. And we develop that at R&D level from 1 to 2 liter fermenter, bench fermenter. Then we have at Fermental also the possibility, a pilot plant, which enable us to grow at 20 liter scale and 1 cubic meter scale. So passing to this fermentation process to the pilot scale enabled us also to start working on the DSP, so downstream process that is summarized here by collecting the biomass, washing it, and then several steps of extraction, to finally end up with the liquid concentrated phycocyanin. And this liquid phycocyanin can be then formulated, dried to end up with the final products. One is called Eversure, and it will be used for coloring foods. And the other one is called Blue Origins, and it will be more targeting the nutraceutical market. As you all know, for phycocyanin as an antioxidant activity, and in our process, we don't end up with the phycocyanin 100% pure. So there's a mix of other protein in the retentate, And we can find also other enzymes such as SOD or catalase that can have antioxidant activity, actually good antioxidant activity. Can you click, please? So this mode of production is can ensure complete traceability from start to finish, because we are growing in sterile condition, and then we do the DSP straight after the fermentation in controlled conditions. On the opposite, the culture of spirulina, of course, is mainly done in open ponds, so it's open to contamination also in the culture. This production for now is it will be done in Europe with different partners, and the advantage also of fermenting this microorganism and that whole process is that enable us to produce the same quality of products all year long, and we are not dependent on the seasons, like for autotrophic cultures. Okay, thanks. Next slide, please. So now I will present different applications that we're developing with Givaudan, and show you what's the difference also between the phycocyanin of spirulina versus Galdiaria. Next slide, please. Yeah. As you all know, maybe or noticed that there's the new, not new food trends, but it's growing and growing along the years. The food industry is switching from chemical dyes or colorants to more natural dyes, and they are used now in candies a lot and other applications. So the main problem for the natural colors is the blue, because there is only one source of blue actually available. That is really Brian blue, and that can compete with the patented blue and the Brian blue, artificial Brian blue. So you have an example here of the other products, natural products that are on the market. But it's apart from phycocyanin, none of them gives real blue colors in beverages specifically, and also in confection. So it's more all purple or blue-gray, but not as Brian blue, not the same hue that the phycocyanin can give you. Can you click, please? So phycocyanin from spirulina, unfortunately, is not suitable for all application in the food industry. There is some problem also of volumes production, consistency of the quality of the product, due to its nature. I mean, the smell of the spirulina itself and the taste of the spirulina. Even the phycocyanin sometimes can still have these off notes, and even with the small quantity used in the product, the final products, depending on the quality you have, you can have these off notes in the final product, which is not suitable for applications. Other issue, it's more or less linked to all the natural colorants, is the problem of thermostability. But also in the case of phycocyanin from spirulina, it's the stability to the pH. And actually, phycocyanin from spirulina lost part of its color at pH under 5 and starts to precipitate around 4.5. That's where we come with an advantage with the phycocyanin from Galdiaria. Actually, phycocyanin from Galdiaria has the same color than the phycocyanin from spirulina, but can stain the acidic pH and never precipitates. So that's why we were trying to develop this product. Is giving a real advantage for the food industry. Next slide, please. Here are a few examples. For example, comparing phycocyanin from spirulina and Galdiaria. In ice cream, they behave the same exactly as a regular confection. we don't have any advantage or disadvantage either. When we start to talk about acidic confections, it's different, as you can see. I hope you can see it with the picture. But the color of the phycocyanin from spirulina is fading with the acidic pH, as I told you before. Next slide. The main advantage we have is in beverages. Here you have the comparison of spirulina, Garderia, and the synthetic blue color. The spirulina, you cannot see it really well on the picture, but you have this, it's kind of, it's not translucent completely, the beverage, and you have a kind of deposit of proteins at the bottom of the bottle, and also you're losing colors. So Garderia can stand the pasteurization also, and in acidic pH, and it's the only stable acid blue that can hold that natural acidic blue color. The blue is not only the blue in the world of the beverages. Can you click twice? We have both of them. If you mix up, sorry, or only once. If you mix them with other natural colors, you can end up with the purple or the green. The green has its importance on the US market because in Europe you can use the chlorophyllene extracted from plants, but the chlorophyllene in the United States is forbidden. So it's more than the blue market, it's a multicolor market, I would say. Next slide, please. So now, as the other people who presented before me explained, we are all trying to improve the sustainability and the profitability of our processes. So that comes in 3 points. That's the main points we are developing at Fermentalg: the process optimization, the sourcing, and the valorization of the side streams. Next slide, please. So starts with the process optimization. It starts with the strain selection, as I already told you, improving the productivity of the strains in terms of phycocyanin contents. So we started from 0 to more or less now 10% of phycocyanin in the dry matter final. In terms of process also, we are playing with the in reducing the time of process in fermentation mainly, and how we can reuse the water in all that processes. Click. Few years back, we also evaluated the possibility of using not glucose, as most of the people are doing, and we're still doing it with glucose, but the opportunity of using side stream for other industry, and in that case, dairy industry with lactose and lactose serum and other products. That the process works perfectly well. I mean, it's just the same as glucose in terms of growth and productivity of biomass. We still have to work on the productivity of the phycocyanin, because you have in the side stream from industries, you don't have always the same consistency of side stream. So that's something we still need to improve to get the same performances with lactose than we have with glucose. Click, please. So now that we have the whole process in extracting the phycocyanin, which is for us the main product now, we also have byproducts. That's the leftover of biomass from which we extracted the phycocyanin. So we are trying to investigate different routes to valorize this byproduct, to call them co-products, and be more sustainable on our processes. So different approaches, the feed, of course, the cosmetics, and some others, but I will mainly present the feed and the cosmetics now. Can you click, please? The first one is the feed industry for feed, aqua, or pet foods. So because we have a specific process of extraction for that acidic resistant phycocyanin, the byproduct we end up is really rich in protein, which is not the case with spirulina, for example. When you extract phycocyanin from spirulina, mostly the byproduct is reduced in terms of protein. In our case, the protein contents even increase during that process, and we end up with like 60-65% of protein in the co-product. It's also rich in different elements, amino acids. Digestibility is pretty good. We have some leftovers of phycocyanin and other elements. To give more values to his byproduct also, we are not looking on the nutritional aspects, but we are also looking for other benefits. So I cannot talk about it right now because nothing is sure now, but we have good reasons to believe that we will succeed in that, but we're still working on it. So click, please. The other approach we had is to see if we could valorize the co-product in other domains like the cosmetic, and we did some work, and actually we have a patent pending on that. And by doing a specific extract on that co-product, we ended up with a product with different activities. One, and that we tested with different labs, well-known labs like Kima and Elysia Biosciences. One was for to test the product on keratinocytes in vitro ex vivo models and proteomic analysis. And we saw that product was improving the regeneration of the epidermis, which is pretty good for cosmetics. And also the same product, when it was applied on specific bacteria like Cutinobacter acnes that provoke the acne, was reducing the growth of those bacteria. Those tests have been done in vitro, of course, and now we are in the process of testing these products to a further level. Next slide, please, and thank you for your attention.
Thank you very much, Olivier, for this really nice presentation. Please ask your question in the chat, keep your question in the room, and we have a question session at the end to answer our speakers all the questions you have. And so we can move on with Mariana Carnero from Necto, and she will tell us about improving the sustainability of microalgae production.
Thank you. Hi, can you hear me well?
Yep. Yes.
Yes? Okay, good. Well, thank you for sharing the slides. And also thank you for inviting me to be a part of this workshop and also have the opportunity to see the other presentations. It's very interesting. First of all, let me give a short introduction. I'm Mariana. I'm an innovation manager and project manager at the microalgae producing company Necton. I will introduce Necton in a few slides, so just bear with me for some time and we'll get right to it. Okay, next slide. As you all know, that is an acknowledge of the importance of sustainability in this day and age, not only individual acknowledgement, but also government-wise. And as most of you know, with increasing global environmental concerns, not only governments, but industries are also pivoting towards more sustainable models. The EU Circular Economy Action Plan, which is part of the Green Deal, is setting the tone for greener solutions in several areas. This includes making sustainable use of our natural resources while promoting a circular economy approach. Next slide, please. As these photosynthetic organisms can provide biomass for a wider range of applications, as we saw in the other presentations, they can alleviate the pressure on less sustainable feedstocks. And this makes them a target for natural, green and blue resources, which explains the growing demand for algae biomass and its derivatives. Next slide, please. However, it's fairly common to use the word sustainable these days, and we have to ensure that we are truly making a progress in improving sustainability. So it's crucial to assess our progress rigorously. There are a few tools for this purpose, most of which you probably already know, starting with life cycle assessment, which is one of the most comprehensive methods being used. Environmental impact analysis that focuses more on the specific environmental risks, and resource efficiency metrics, which allows us to measure the inputs like energy, water, and nutrients. And by using these tools, we can quantitatively assess our progress towards a more sustainable microalgae and macroalgae production to make data-driven decisions. Next slide, please. So there are several strategies to balance this sustainable production and cost, of course.
Sorry?
Oh, no, before. So this involves different strategies, some of them which were already mentioned before, such as, for example, strain improvement, side stream valorization, and biorefinery. We have also...
It's getting itself, I don't know.
Oh, I'll try to be fast. So also optimizing input usage, recycling nutrients, reducing energy requirements, while of course always ensuring that productivity stays high, which is also a requirement for our production. Should I stop just to check if everything is okay, or?
I don't know if it has a timer, but it seems that the slides have timers. Yes.
Okay. I didn't know this, sorry. Okay, so I'll try to to be on time then. So now I'll briefly give you a presentation of Necton as promised. For those of you that don't know, Necton is a microalgae producing company located in the south of Portugal, in Algarve. It began as a spin-off from the university in 1990, and it was officially founded in 1997. Currently, Necton is one of the oldest microalgae producing companies in Europe, which is still in operation. And we currently produce for different sectors, but mainly for aquaculture and cosmetics. Next slide. We have always tried to improve the sustainability of microalgae production through intensive research and continuously staying updated with biotechnology innovations. This led to the participation in different national and European projects that you can see here. Fortunately, with many happy partnerships with different universities and industries and non-profit organizations. And so I will present some of the strategies that were involved in this, in some of these projects. So here it's the AlgaVlor project. We used artificial lighting, so LEDs, to reduce the nighttime biomass losses. As you can see from this graph, we were able to sustain biomass growth even during the night periods by supplementing light through energy efficient LEDs. However, artificial lighting is cost effective only for high value products, which reinforces the importance of maximizing natural sunlight or use renewable energy to power these LEDs. Maybe I was trying to see if I could find the timing. I don't know how to take it off. So yeah, this one. So in another project, Magnificent, we increased the volume of the PBRs. You can see in the picture, the left was before and the right PBR after we increased the volume vertically to maximize the biomass production per area. In the graph below, you can see the growth of both tubular photobioreactors, 15 cubic meters and 20 cubic meters on the right. The growth was not impaired by the added volume, so it could produce more biomass per square meter without losing productivity, which translates in a more efficient use of the land. Next slide. Still in the same project, we also try to decrease energy consumption. Energy used to mix algae cultures represents a major economic and environmental cost. And from previous LCAs done at Necton, we knew that the pumps had a great energy demand. So we grew microalgae in these tubular photobioreactors while shutting down the pumps during the night when no photosynthesis occurs and restarting the pumps after 8 hours in the early morning. As you can see in the plot on the right, we observed similar growth performances between the growth and our treatment with the pump off for 8 hours during the night, meaning we could save 1/3 of the energy used in the pumps. Next slide, please. This is another project, ProFuture project. It's Horizon 2020, where we continue to develop this approach. Since renewable energies are key to improve algae production and sustainability, we built a tubular photobioreactor which could work only with the energy generated by the photovoltaics that cover Necton's rooftops, as you can see here on the right. We already performed several tests growing Nonochlorops oceanica for more than a month, using only the energy from these photovoltaics, meaning if the light was limited by nighttime or dense clouds, the pumps would be off. In the plot below is an example of when we tested this concept with 2 different setups. We started by forcing the pump to work for 5 minutes per hour, regardless of having enough light from the solar panels. This is represented by the gray areas of the plot, and then switched to have the pump completely off for another week if there was not enough light, which is represented by the shaded yellow area. These 2 setups were alternated for a month to evaluate biofilm formation and limited growth, but no obvious differences were observed, meaning we could operate the PBR with the photovoltaics alone. The next slide. In the same project, we also assessed biomass drying, which is a very energy demanding step as well. We wanted to assess if solar drying as a sustainable drying technique for microalgae industry. So we compared different drying equipments by evaluating biomass composition between freeze drying, solar drying, and spray drying. In the plot, you can see the fucoxanthin content of Phaedacnum tricornutum dried in the different systems. And although the solar dryer biomass did not reach the gold standard of the freeze dryer, it did surpass the quality of the spray dryer biomass, revealing the potential of the solar dryer even for high value applications. As such, solar dryer offers a more sustainable alternative without drastically compromising product quality. Also in the same project, inorganic carbon is, as you all know, key for industrial cultivation of phototrophic algae biomass. And as such, there are already several studies on capturing CO2 from polluting industries to use in algae production facilities. A recent approach is the use of in situ direct air capture systems for CO2 absorption and concentration from the air. So the University of Twente, which is a partner from several projects, is working on the development of a direct air capture prototype system to deliver. It's okay. 10 kilos of CO2 per day.
I'm just trying to fix it. I tried in the other computer.
I was trying to see if I can find. I think I can, in the slideshow tab, in the setup, there is a record button. And if you click it, there's a clear timing on all slides. Maybe that's it. I don't know if you could follow. On slideshow setup,
Yes.
record.
I'm just sharing the screen now. 5 seconds, please. And just take it out of there. I believe it's right now. Can you continue? Do you see the presentation?
Oh, sorry, I was muted. Yes, I can see it. Sorry. Yeah, I was just sharing that this direct air capture system is being optimized to improve the current productivity, which is 10 kilos of CO2 per day directly concentrated from the air. If you want, if you take it out of presenter mode.
It's okay now.
It's okay? It was you. Okay. Okay. All right. So now on another project about the massive quantities of marine and fresh water that are necessary, as well as inorganic and organic nutrients, such as nitrogen and phosphorus. We know that they have a great impact in the environment, and the recycling is one of the many focuses of the European Green Deal in different industries. So the reuse of water and nutrients from adjacent industries. We saw also in the previous one, like I think it was the dairy industry. In this case, we use the agricultural industry to provide rainwater to serve as an interesting source of water and nutrients for microalgae growth. And for this, we tested to grow the microalgae in this rainwater and clean the water while producing microalgae biomass, which was biorefined to produce 2 different types of products, biostimulants for agriculture and aquafeeds for aquaculture. Next slide, please. Here you can see the inoculation in this project of Tetradesmus obliquus in a tubular photobioreactor filled with this agricultural drainage. Just a click, please. And on the right, you can see the growth of this algae together with the nitrate consumption below, which reached below the legal limit for discharge. Okay, next slide. So with all of this in mind, we have a more recent project, which is called Realm, which was funded under the same call as CIRCALGAE, so they are sister projects. And here we wanted to implement several of these strategies that we already tested at lab or pilot scale. And here we have a bigger plant-sized demonstration facility where we aim to, first of all, move the microalgae production next to key inputs of water and nutrients, such as the greenhouses of farmers. And here produce the algae using resilient strains and an automatic operation powered by, through autotrophic production, concentrated in situ, and click please, and moved to a centralized processing facility to have a multi-product biorefinery for product development, for products for aquaculture, such as specialized feeds and agriculture as biostimulants and biopesticides. Next slide, please. So the end message is that there are many approaches that we can take to optimize microalgae production. We should integrate different approaches, biological and engineering as well, to try to really reduce technical bottlenecks of algae cultivation and improve the environmental benefits associated with algae cultivation to have a better management of natural resources. So this is just a piece of the puzzle. Of course, we still need a lot of work. We still have a lot of work ahead. But yeah, I think with all of this research that we see these days, it's important to integrate and share the knowledge. Since we have a long way to go comparing with the agricultural products as it was shown. And I think that's it. Thank you much. Thank you very much for your attention. And I hope you enjoy the rest of the seminar.
Thanks a lot, Mariana, for your presentation. We keep questions for after the last presentation of this session. And so we open the floor to Jean-Eric Jessen from AlgaLeaf. He will tell us about astaxanthin business.
Thank you very much. It's a pleasure to join you on this webinar. Do you hear me okay?
Yes.
Good. So my name is Jan-Erik. I'm head of R&D at AlgaLeaf, Iceland, and I'm going to tell you a little bit about our company and do sort of a recap from a 10-year journey in the astaxanthin business. So if you can move to the first slide, thank you. So first, some historical context. AlgaLeaf started off as an innovation project in Norway, and it was bought by individuals with experience from the nutraceutical industry, and they believed that this project could be developed into a profitable biotechnology company. So the initial method development took place in Norway, but for several important reasons, and I will get back to that later in our presentation, it was decided to relocate the company to Iceland and build the factory here. So this was done in 2013, and then the first phase of the plant was constructed. And then 1 year later, in 2014, the company started to commercially cultivate the microalgae Haematococcus pluvialis for the production of astaxanthin. So next slide. So this is a picture of our headquarters in Ásbrú, Reykjanesbær, and I want to point out to you that it's located in only 5 minutes driving distance from the international airport. So if you ever come to visit Iceland, I suggest to you that you should pay us a visit and have a look at our facilities. We're always keenly welcoming guests to our plant. And next slide. So you might have heard or not on your news that there is a lot of volcanic activity in Iceland currently, and I just wanted to show you this map to highlight that the distance from the active site to our company is around 12 kilometers. So that might sound a little bit scary, but it's just we call this eruption like a tourist eruption, because you can almost just walk up to the site and have a look at it with your eyes, because it's fairly like yeah, not too violent. It's pretty calm, let's say. And if you go to the next slide, you can see I took a short video a few days ago, or a few weeks ago actually. So next slide. Thank you. So this is our headquarters that you see, and if if you press enter once, then the video should start. Lagging a little bit, but this shows you basically how close this eruption is to our plant. All right, and then we move on with the presentation. And let's stop talking about eruptions and start talking about microalgae. So first, I wanted to talk to you a little bit about this compound, astaxanthin. So it's a ketocarotenoid compound that the microalgae is producing. And it's one of the strongest antioxidants in nature. And as a nutraceutical, it has a wide range of health benefits for human. I'm not going to discuss this in any detail, but among those are benefits for your skin, eyes, joint health, cardiovascular health, muscle recovery, and so on. And the reason why this range is so wide is basically that as an antioxidant, this has a very strong or a broad effect on so many systems in the human body. Astaxanthin is also a deep red colorant, and you will know this compound from salmon or shrimp, for example, because this is the compound that's responsible for the red color that you will see in salmon or shrimp. So astaxanthin can be produced in two different ways, either naturally, as we do by cultivating the microalgae, or synthetically from petrol oils. Only the natural form of astaxanthin is accepted for human consumption, but the synthetically produced form is mostly, or is by far, what they use in the salmon farming industry as a colorant for the salmon meat. So in nature, this freshwater algae, Haematococcus pluvialis, is the organism that can produce astaxanthin by far the highest concentrations. And that is why natural producers of astaxanthin are mostly cultivating Haematococcus pluvialis to provide the compound. So the dry weight of astaxanthin can exceed 7% of the dry weight of the biomass. So this is how good the organism is in creating this. Okay, thanks. You can go to the next slide. So we cultivate our microalgae indoors in closed tubular systems with artificial illumination. It has its pros and its cons. We'll see if I'll have time to discuss that later on, but I just wanted to point to you at this point that this is the way that we cultivate here in Iceland. You could imagine that it's hard to cultivate microalgae outdoors in such a cold country. So this is our approach. And the next slide. If I go just very briefly through the production process, we start by taking microalgae material from slants and agar plates and inoculate very small flasks. And let's say the first half of the cultivation process is just to scale up the volume of the algae that you need, going to bigger and bigger and bigger volumes. And then there in the middle of the picture, you see that we change our approach. We change the approach from trying to provide the best cultivation circumstances possible over to trying to get the algae to produce astaxanthin. So in nature, this microalgae reacts to unfavorable environmental conditions by starting to accumulate astaxanthin, which protects the algae from this bad environment, too much heat, too high salinity, no nutrients, and so on. And then waits until the circumstances get better, and then they release the astaxanthin and start growing again. But in our case, that's not going to happen because we are, of course, harvesting the microalgae when it has accumulated a lot of astaxanthin. So what we do at the beginning in this intermediate, what we call starvation phase or brown phase, is that we stop feeding the microalgae with nitrate. We elevate the temperature around 4 to 5 degrees above the optimal temperature for growth. And we also change the light composition. In the green phase, we have mostly red lights, a little bit of white, but we increase the light, the white proportion, and decrease the red to provide light that is in lower wavelengths, more energy, and this provides a lot of stress to the cells. So the green phase takes around 7 days or so, the starvation phase maybe 4 to 5 on average, and then we transfer the cultures to the red phase, and there basically we throw in all the stress factors that we know of. So there we also add salt and iron and change to blue lights with very low wavelength and very high energy. And all this combined makes the algae produce a lot of astaxanthin. Then after 9 to 10 days on average, it's time to harvest. Then we decant or get rid of the water. We will have to use a ball mill to rupture the structure of the cell so that the astaxanthin gets more accessible. And then finally we dry. The dried microalgae biomass contains around 6 to 7% astaxanthin, and some of our customers buy the biomass, but the majority we have to provide the astaxanthin extracted in the form of oils. So this is done with critical CO2 extraction. Okay, then the next slide. Then you might ask why producing astaxanthin? And the obvious answer is that this is, of course, a very valuable product. Every kilogram of pure astaxanthin is worth maybe around $8,000 to $10,000. The market size of natural astaxanthin as a nutraceutical is estimated to be around 35 tons this year, and the growth is estimated to be maybe 10 to 15% annually. So there is a big potential for growth in this industry. Then there are also some opportunities for astaxanthin in other markets, which we are always exploring. First one being in the feed market, because there is increased interest in providing organic cultivated salmon, and for that you would need natural astaxanthin, not synthetic as is mostly used. So we have had interest from some companies about this. Then we have been approached by very large companies in the human food sector that want to use astaxanthin for certain types of food. And then last but not least, there are opportunities for the API applications. We are currently working with a company or a research group that is developing a certain type of medicine against certain liver diseases. Of course, astaxanthin alone will never be accepted as medicine, but it can be one of the active ingredients, and the antioxidant and anti-inflammatory properties of the compound, they make it interesting for medicine development. So, but this is of course like the long game. It takes a lot of time to develop a new type of medicine. So we are aware that this is not going to happen maybe in the next year or next 2 years, but sometime in the future we believe that astaxanthin will be a part of a medicine. Next slide. And then you might ask why we should be doing this in such a small country that is so far away from all the markets. You have the earthquakes, you have the volcanoes, you have the highest salary costs in the world. Maybe Iceland and Norway are together there in the first place of the highest salary costs. So why does it make sense to do this in Iceland? So I want to go through these few advantages that Iceland has as a company for this kind of an industry. So first of all, and maybe the most significant part, is the sustainable green energy that we get from hydraulic and geothermal sources at very low prices. So the energy costs for us, if we would be located in somewhere in Central Europe, for example, we would be expecting to pay 5 to 6 times more for the electricity than we do today. And electricity is a significant part of our production cost because of the artificial illumination. The LED lights, they are very energy efficient, but they still consume a lot of energy. Then we have access to clean fresh water in abundance, so to say, free of charge. And this is very important because we need a lot of fresh water in our cultivation. And I also have to mention that the cold and not so nice weather is actually very positive for us, because it makes it easier for us to cool down our facilities. So even though we're located in a country where you might expect -degrees in the wintertime, we still need to put money and effort into cooling down our facilities, not heating them up, because the LED lights, they release such amounts of temperature. So that's why we have to cool them down. And then also, what you can also say about the temperate climate is that it provides less bioburden, because you have not as many species of contaminants that can thrive in the environment around us, and that means that it's easier to keep the cultures clean from contamination. And next slide. So what I said before is that we relocated to Iceland in 2013 and started commercially producing in 2014. But we expanded in 2018 and 2019. Then we implemented what we call the phase B of our plant. So then we went from 400 kilograms of pure astaxanthin annually to around 1,500. And now we are actually in another phase of expansion, implementing what we call phase C. And after that, we will be able to produce around 5,000 kilograms of pure astaxanthin annually, which will make us one of the largest producers of natural astaxanthin in the world. And at the same time, we are installing a critical CO2 extraction facility on site. So we don't have to get that contracted anymore, because the critical CO2 extraction is a very complicated process that requires very expensive equipment, and it's very expensive to contract that kind of a process. So we will be more competitive after installing our own critical CO2 extraction facility. And the next slide. So here are just a few pictures to show you this. You can see top left. This is basically the expansion consists of 2 buildings. One of them is a greenhouse. And that's what you see top left. They're basically the 2 later stages or 2 last stages of our cultivation take place. And then we have a steel building, which you see. Maybe it's not very easy to see it, but on the second picture to the right, it's the steel building, and there you have the laboratories, the offices, the warehouse, the downstream facility, and basically the rest of what's needed for the process. And what you can see also bottom right, sorry, that's the new critical CO2 extraction facility. And the next slide. So to the topic of this presentation, what have we learned from this 10-year journey? First of all, you have to play to your strengths. So Algaleaf is always considering options of cultivating other microalgae, but for like to produce other products, but they will always have to be both phototrophic, because we need to be utilizing the inexpensive energy that we have to be able to compete with bigger countries with lower salary costs. And also we have to be cultivating, or we have to be producing chemicals that are very valuable and very small. And then for other areas in the world, a completely different scenario might be the case. But this is our reality. Then focusing on cultivating one particular microalgae for production of one particular compound is a good strategy in the short term. So basically, we became very good in cultivating this particular microalgae, establishing the company by being able to produce this compound very effectively. But in the long term, we will always have to look at other applications and see if we can start commercially cultivating other microalgae, because when you're only like cultivating one microalgae, you basically have all the eggs in the same nest. We started off trying to build up our own brand, and we have our own brand, but in a niche market like this that already exists, it's very hard. So we soon came to know that we would be able to produce much more astaxanthin than we could ever sell ourselves. So 95, 96, 97% of what we produce is sold B2B to other businesses, to other companies that use the compound for their brands. When it comes to productivity in microalgae cultivation, the artificial illumination, or the illumination in general, is, we would say, the most impactful factor. We know this because we have been spending a lot of time and effort into developing our lights, and we can see that by optimizing like the wavelengths, the intensity of the light, it can have a very, very significant impact on the productivity of the cultivation. And then if you're oh, sorry.
Sorry. I was sending a message.
Just 2 more points and then I think I promised I'm very close to the end. So if you're cultivating a relatively slow growing microalgae in photobioreactors under mild conditions, as we do, meaning that the temperature is close to room temperature, the pH is close to neutral, and when you have these mild conditions, it basically means that contamination is always going to be a big challenge. And I guess every producer of microalgae in at least in tubular photobioreactors knows this. So what we have come to know is very important for this process is to be very good at analyzing your cultures, detecting contamination, reacting to it as quickly as possible, and basically learning a little bit to live with at least certain types of contaminants. Because no matter how clean your processes are, there is always something that's going to enter your bioreactors and contaminating them. So you have to be very good at managing those situations to provide as clean cultures as possible. And then, of course, last but not least, and basically every company must have this experience that skilled and determined staff is key to success. And I don't think I need to explain that in any more detail. So I'm 2 minutes over time. Thank you for this, and happy to answer questions. If the time does not allow everybody to get answers to the questions, please just contact me. I guess my email address I think is on the last slide. No, it's not there. But you can just easily look me up at algaleaf.com, and please contact me if you have any questions or if there's anything you want to discuss. Thank you very much for your attention.
Thank you, Jean-Eric, for this presentation. And as you said, we can now start the question session. Are there questions in the room first, maybe for one of our speakers on the 2 session? I don't know. If not, it's not a problem. I think we have a lot in the chat too.
Yes, we have some questions to you, John, in the chat. I will start from the bottom to the top. So first, Bartholomeus, if I'm not, please, I'm reading the names. I don't really know everyone. Asks if astaxanthin production are known for being reluctant to fungi contamination. Do you face similar condition in your closed and indoor cultivation?
Yeah, I mean, we see all kinds of things, of course. If you just examine all our cultures, there are all kinds of things that we have seen over these 10 years. Fungi is something that we have sometimes seen, but that is not like, I would not say that fungi is like a severe contaminant to us, because when we have seen some fungi in our systems, what they normally do is they go up to a certain point and then they eventually start to fade again. So we have not had any like severe fungi contamination problems. What is always the most severe type of contamination to our cultures is other types of microalgae. And the reason for this is because we are providing such good cultivation environment for microalgae that if we have some microalgae contaminant that grows faster than Haematococcus, it will eventually take over the culture at some point. So this is basically at least our experience with the contamination.
Thank you. And also Olivier asks if what are the regulation barriers for astaxanthin as colorant for human food, for food?
Yeah, that's a question that I would have to take a significant amount of time to answer. I think it's maybe not easy to answer this very briefly, but if I make an attempt, astaxanthin is of course a fat-soluble compound, so you cannot consume astaxanthin in high amounts without having some kind of accumulation in your tissues. So it means that you cannot add, according to regulations, you cannot just add astaxanthin to food So basically today it would not be possible to add astaxanthin to food in any significant amounts because of this fat-soluble property that the compound has. So we have been in discussions with some companies about adding astaxanthin to food, but it's never really come to the point where we are close to putting something to the market, at least not yet. So, but yeah, I could discuss this like in much more detail, but this was my very short answer to the question.
But this is my question. If this is something that addressed just to the European market or other markets, you'll see different kind of opening or closed, environment to address this kind of product.
Yeah.
Well, there's another question also, if to reach 7% of astaxanthin in the biomass is achievable.
Yes, it is achievable. We have exceeded 7%. Of course, it's not every batch is that good, but of course our algae is not genetically modified. It's just natural microalgae. But what we have done is basically the same thing as people have done in agriculture for tens of thousands of years, to basically take the best individuals and try to cultivate them further. So there might be some genetic drift some modifications, some mutations in the microalgae that make them better than other strains in producing astaxanthin. But yes, we have on a regular basis, we exceed 7% in a batch.
Thank you. For this second session, we have included
Okay, I have a question to Olivier. Can you tell us, or do you know why there is such difference in stability between phycocyanin from spirulina and Galderia? Is it due to the process of cultivation process, or is there different, I don't know, molecule structure that explain this difference of stability?
Good question. No, I mean, we're not sure. I mean, we just have the hypothesis on that, but it's what we think it's that it's mostly due to the protein part of the phycocyanin and its structure and its stability toward temperature or acidic or things like that. But difficult to, I mean, really answer that question because it's only hypothesis, and structurally they are really, really similar, both of them. Only the amino acid sequence is changing in few places. And that's our explanation. And I know that there's other articles that has other explanations, but there is no clear answer on that. But the fact is that it's not only Galderia sulfuraria, but it seems that red algae have the same specificity.
Thank you. Is there a question for the first session?
Yes, for the first session, we have actually questions to Marco.
Okay.
Yes, if you would like to come here, just to, because I want to be close to the microphone. So Marco, there's one question from Romain. What are the targeted production costs and OPEX and CAPEX for dry biomass obtained from autotrophic and heterotrophic processes in order to be competitive against alternatives? Is there an answer for that?
Maybe there is, but
You have an hour.
I really don't have this data.
Yeah, no, and I mean, depending, I mean, what is the alternatives? I mean, this is a very complex discussion and comparison how to do and which kind of markets, because even if you, I'm saying even if you focus on the cosmetic market, you have cosmetics from upper prices and lower prices. So it's very complicated to do a simple answer for that.
So for every application, for sure, it depends on the alternative that is already on the market. So
yes, I know.
I remember my first project that I did was about fish feed, and I had to compete with soybean protein. So it's like cents per kilogram. And to reach there with algae was very hard. So like I have these comparisons, but for cosmetics, we can have higher prices because we put
You
less.
can find, yes, a better where to fit your product. Well, and another question is, how are the contaminations at the open fund system pilot plant being handled?
We cultivate in closed,
yes,
tubular photobioreactors, but still we have contamination sometimes, amoeba, for example. So we try to go for a high pH.
Je pense aussi c'est qu'on a quand même des taux de. Ah, on n'a pas fait le clarifiant en mesure pico.
Sorry. Okay.
Yes, basically, if a culture is contaminated, we have to discard it and clean and disinfect everything. So there are very few strategies for algae available that will target the contamination and have like a contrast strategy against it.
Okay, another question is there any study comparing natural sunlight versus artificial light that can produce different results? And maybe not just for you, but if someone else would like to also respond, and which one can produce better quality of product output? I mean, better quality is always something, what is to be better, but depending on what your goal. But
for sure, for sunlight, the quality is changing a little bit more than you have in reproducible conditions in the artificial light. But we didn't see so far a worse quality in one of them. We saw now in the CIRCALGAE project, Pheodactylum, for example, in all of the artificial light cultivations, we had a much higher chlorophyll content, which was unwanted in our case. So it was not better quality from artificial light, but we still have to reproduce it, and we plan maybe next year then to make a publication on that.
And a last question is, what is the dry weight and fucocyantin content in your cultivation of Phytact?
So for Pheodactylum powder, after drying, we have usually 0.8 to 1% of fucocyantin that we measure in the dry powder after freeze drying. like we are in this range, not much higher, not like the 7% that we heard for astaxanthin. Yeah, and
So
for the dry weight content of the culture, we try to go as high as possible to have this problem of the medium a little bit minimized, because we use saltwater medium, and even if we try to recycle, we have to go as high as we can. But as soon as we see the growth curve is flattening, we lose then growth speed, and we have to harvest partially. And yes, in the bigger tubular photobioreactors, this can be at 3 grams per liter. We can reach even higher concentrations, but then at a lower growth.
okay, so that were the questions that we had already. It's still open. If I don't know if someone else would like to make any other questions. And
Yes, me,
Thank you,
I have one. Thank you, Marco, for your answer. But you can stay with me if you want. I have one question for producers of microalgae, so maybe A4F, Necton, IGV, I forgot someone in the speakers, but we talk about sustainability today and best practices, but what are the main challenges to have sustainability, but also suitability and profitability in the system, and what, in your opinion, is the main obstacle to be lifted? Louis.
Big question.
Just part of, just your feeling on this subject, because we talk about a lot about sustainability, but the main objective of company is profitability. So
stay here.
Sorry.
Everyone join us.
Getting close to the microphone. That's a very big question. So today, I think it was evident that we saw very many different cases of microalgae producers, and the technologies, the species, the conditions, they were all different. So that means also that the sustainability assessment is different also in all these cases. So it needs to be done individually. So that makes it harder to compare microalgae production with something else. So it needs to be on a case-by-case assessment. And also your question to me, it also makes me think about something that actually Joana Valls from Necton mentions quite often, and I will repeat it, which is also when we assess the sustainability of microalgae against sustainability of alternatives, are we really accounting for the sustainability issues in the alternatives? Are we paying the price of the lack of sustainability of some of these alternatives? If we were to impose the same criteria to the alternatives, would their cost be what it is today, or would it be different? So to assess, to compare these two things nowadays, putting microalgae to these tough tests that only come in these recent decades. And all the alternatives have been there for 1 century, and no one was really too bothered about sustainability issues. It's a very different barrier to overcome for the microalgae business. So if that comparison becomes more reasonable, I think that this will bring about increase of opportunities for the market of microalgae, which is necessary. And I think the sustainability assessment, doing the product environmental footprint and all the LCAs assessment to these products that are derived from microalgae will help put these things in perspective to analyze in a more fair way microalgae products and their alternatives.
Yes. And it makes the link with what Marco is saying about agriculture and the way we have to do to reach the same developments, and we are in fact at the beginning.
Technological path and innovation curve that we have to go through. And there's last question, but also connected somehow to Luis, that is Gabriel also asks that, can you say something about which nitrogen-rich effluent sources you tried? So we have also examples here of side streams, thinking about side streams. So our raw material comes already from a byproduct or side streams from others. This is already a sustainability criteria.
So in the particular case that I showed, the two different sources were tested. One of them is coming from the chemical industry that is adjacent to this microalgae cultivation facility, and it's a side stream from that base chemical manufacturing process, which is rich in nitrogen. And the second one is from an industrial facility that is just maybe not even 5 kilometers away from this facility, and it's coming from a fertilizer production plant, and it is also a liquid effluent that's rich in nitrogen that is a problem for them, and it is can be used as a raw material for algae cultivation.
Thank you.
Okay, thank you. I think it's time to close this event. Before, I would like to thank a lot A4F for hosting this event, to all the people of A4F for the organization too, to all the speakers for maybe questions that haven't been answered, or if you have questions for our speakers that haven't take the microphone to answer, feel free to contact them or to contact us, and we can share your question to speakers. So thanks a lot to all of the speakers, for all the attendees of this event, and to all of the CIRCALGAE also partners that have stayed here this afternoon with us. Just stay tuned, because there is another workshop webinar next year in the CIRCALGAE project, more on the biorefinery aspect. So you can stay connected to the CIRCALGAE network, and CEVA and Aquimer and all partners in the CIRCALGAE project for the next step and the next training and next workshop. Thank you.
Thank you.
Thank you very much.